Welcome to Biology, the scientific study of life. RNA plays multiple roles in the cell. The cellular machinery of protein synthesis and RNA targeting is dominated by RNA of various kinds. In addition to mRNA, this includes tRNA or double RNA, and in eukaryotes, snRNA and srpRNA. This table, notice that a type of RNA called small nuclear RNA. Notice that a types of RNA in eukaryotic cells. Type of RNA, messenger and functions. Notice that messenger RNA between two brackets mRNA carries the functions as carries information specifying amino acid sequences of proteins from DNA to ribosome. Transfer RNA T between two brackets tRNA, the function as serves as adapter molecule in protein synthesis, translates mRNA codons into amino acids. Ribosomal RNA between two brackets R double RNA plays the function as plays catalytic ribosomes roles and structure roles in ribosomes. Boom. A type of RNA primary transcripts. The function is serves as a precursor to mRNA, double RNA, or tRNA before being processed by splicing or cleavage. Some intron RNA acts as a ribozyme, catalyzing its own splicing. Boom. Type also of RNA, small nuclear RNA between two brackets is in RNA. The function is plays structure and catalytic roles in spliceomes, the complexes of proteins and RNA that splice pre-mRNA. Also, the type of RNA is srpRNA. The function is a component of the signal recognition partial SRP, the protein RNA complex that recognizes the signal peptides of polypeptides targeted to the ER. Also, the type of RNA, small nuclear RNA, is in RNA. The function is aids in the processing of double RNA transcripts for ribosome subunits formation in the nucleus. Also, the type of RNA, the last one, small interfering RNA, siRNA, and microRNA between two brackets miRNA. The function is are involved in regulation of gene expression. Okay. Comparing gene expression in prokaryotes and eukaryotes reveals key differences. Although prokaryotes and eukaryotes carry out transcription and translation in very similar ways, we have noted certain differences in cellular machinery and in details of the process. Prokaryotic and eukaryotic RNA polymerases are different. In those of eukaryotes depend on a complex set of transcription factors. Transcription is determined differently in the two kinds of cells. Also, prokaryotic and eukaryotic ribosomes are slightly different. The most important differences, however, arise from the eukaryotic cells compartments organization, like a one-room workshop. A prokaryotic cell ensures a streamlined operation. In the absence of nucleus, it can simultaneously transcribe and translate the same gene, and the newly made protein can quickly diffuse to its site of function. In contrast, the eukaryotic cell's nuclear envelope segregates transcription from translation and provides a compartment for extensive RNA processing. This processing stage provides additional steps whose regulation can help coordinate the eukaryotic cells' elaborate activities. Finally, eukaryotic cells have complicated mechanisms for targeting proteins to the appropriate cellular compartment, which means organelle. Where did eukaryotes and prokaryotes get the genes that encode the huge diversity of proteins they synthesize? For the past few billion years, the ultimate source of new genes has been the mutation of pre-exciting genes, the topic of the next section. Bon. Notice this figure coupled transcription and translation in bacteria. Notice this line, so fine. Notice that, so fine. RNA represent RNA polymers. This will also represent polyribosome, and this one DNA. So fine. Notice mRNA. Notice here. Okay, a closer look, or this one is DNA and polymer RNA polymers. Notice that closer look polyribosome, and closer look this one polypeptide amino acid. Each one is polypeptide amino acid, and this one also ribosome. The red one is mRNA 5' here. And also notice that in prokaryotic cells, the translations of mRNA can begin as soon as the leading 5' of the mRNA molecule peels away from the DNA template. The micrograph using TEM shows a strand of E. coli DNA being transcribed by RNA polymers molecules. Attached to each RNA polymers molecule is a growing strand of mRNA, which is already being translated by ribosomes. The newly synthesized polypeptides are not visible in the micrograph, but are shown in the diagram. Notice that. Point mutations can affect protein structure and function. Let's talk about mutations. Mutations are changes in the genetic material of a cell or virus. In this figure, notice that we considered large-scale mutations, chromosomal rearrangements that affect long segments of DNA. Now we can examine point mutations, chemical changes in just one base pair of a gene. If a point mutation occurs in a gamete or in a cell that gives rise to gametes, it may be transmitted to offspring and to a succession of future generations. If the mutation has an adverse effect on the phenotype of an organism, the mutant condition is referred to as a genetic disorder or hereditary disease. For example, we can trace the genetic basis of sickle cell disease to a mutation of a single base pair in the gene that codes for one of the polypeptides of hemoglobin. The change of a single nucleotide in the DNA template strands leads to the production of an abnormal protein. Notice this figure. In individuals who are homozygous for the mutant allele, the scaling of red blood cells caused by the altered hemoglobin produces the multiple symptoms associated with sickle cell disease. Let's see how different types of point mutations translate into altered proteins. Types of point mutations. Point mutations within a gene can be divided into two general categories: base pair situations and base pair insertions or deletions. While reading about how these mutations affect proteins, let's talk about this figure and more about base pair substitutions. A base pair substitution is a replacement of one nucleotide and its partner with another pair of nucleotides. Some substitutions are called silent mutations because owing to the redundancy of the genetic code, they have no effect in the encoded protein. In other words, a change in a base pair may transform one codon into another that is translated into the same amino acid. For example, the 3-CCG5, notice that, on a template strand mutated to 3-CCA5, the mRNA codon that used to be GGC would become GGU. Notice that instead. And a glycine would still be inserted at the proper location in the protein. Notice that. Other substitutions may change an amino acid but have little effect on the protein. The new amino acid may have properties similar to those of the amino acid it replaces, or maybe in a region of the protein where the exact sequence of amino acids is not essential of the protein's functions. However, the base pair substitutions of greatest interest are those that cause a readily detectable change in a protein. The alteration of a single amino acid in a crucial area of a protein, inactive site of an enzyme, for example, will significantly alter protein activity. Occasionally, such a mutation leads on an improved protein or one with novel capabilities, but much more often such mutations are detrimental, leading to a useless or less active protein that impairs cellular function. Substitution mutations are usually missense mutations, that is, the altered codon still codes for an amino acid and those make sense, although not necessarily the right sense. But a point mutation can also change a codon for an amino acid into a stop codon. This is called a nonsense mutation, and it causes translation to be terminated prematurely. The resulting polypeptide will be shorter than the polypeptide encoded by the normal gene. Nearly all sense mutations lead to non-functional proteins. Okay. Back to this one, the molecular basis of sickle cell disease, a point mutation. The allele that causes sickle cell disease differs from the wild type normal allele by a single DNA base pair. Okay, thanks for listening.